Hi, everybody. Happy Monday. It is 8 o'clock, and you're here with Kari Parnan. I'm one of the biology Fiveable streamers. So if this is your first live stream event, welcome. It looks like we have a couple people here already. So hi, Sandra. How are you? So if you want to say hi, go ahead in the chat box. Why don't we go ahead and share just real quick what you're learning about. Like, what did you learn about in biology today? If you had biology today. I know some of you might be on a block schedule. But go ahead and just type in the little box right here what you learned about today in AP biology. Just so I can see where we're all at in terms of learning. Oh, nervous system. Okay. Okay, so Soleil's working on nervous system in their class. Who else? If you guys want to just share what you're doing, that'd be great. If you learned about, some of you might even be on natural selection already, or genetics, DNA, cell cycle. That's kind of where we are going pretty soon with the next unit. Will be unit 4, and we'll be looking at cell cycle, why a cell behaves the way it does. You have it tomorrow. You have a test on the membrane structure. Okay, so you guys are on cells still, Sandra. Okay, membrane structure. Good. Okay. All right, so we have about 6 people so far, 7 people now. Welcome. Thanks for coming to the Fiveable Unit 3 review. I kind of have organized this similar-ish to a college-style review session. So I honestly want you guys to tell me what to talk about. I have a couple things planned in terms of I have a couple free responses that we can go over and talk about how you would answer them correctly. But other than that, I mean, I really want this to be in your hands in terms of what you want to talk about. So just to remind you, think Fiveable if you haven't already. Just kind of a heads up. Unit 4 is next, so that's everything about the cell cycle. Also, you'll be hearing about some changes. At Fiveable, we'll be adding some more practice exams, practice questions, things like that, other than live streams. So it'll be really, really cool coming up soon. Today, we'll do some practice free response questions and then open forum. So I think I might flip it actually and have the open forum first and have you guys tell me what you want to talk about first. But just to remind you what Unit 3 was all about, Unit 3 is cell energetics, so how a cell takes in and uses energy. So most teachers would have started talking about enzymes, like what is an enzyme, how does it work, how are they inhibited, how are they activated, what affects them, cellular energy, things like that. And then it would have ended up with cellular respiration and photosynthesis. So I know those are kind of the big heavy hitters that everyone gets confused on. So with that said, why don't we go ahead and take a second and think. And then if you can, please type in the chat box, or you can use the Ask a Question button down here. It'll pop up. You can hit that and type in your question, and then everyone else in the stream can kind of vote to whether or not they'd like me to go over that. Because honestly, if we don't have time, I'll kind of pick the ones that were most heavily voted. So take a second, and if there's anything about enzymes, cellular respiration, and photosynthesis that you want me to answer, it could be a specific question, or it can just be, hey, can you go over glycolysis? Or can you go over something specific? Don't just say go over photosynthesis, because you can watch the previous live stream on that. Okay, we want to go a little more specific with what you want me to go over. So go ahead, take a second, either type it into the Ask a Question button, or go ahead, you can just write it in the chat box, either way. So go ahead, I'll be waiting on you guys to type in a question that you have about Unit 3, or you can write it in the Ask a Question poll. So your question could be, could you explain, enzyme inhibition, or what's the difference between competitive and non-competitive inhibition? Or you could, there's so many questions you could ask. So I'll give you all a minute. This is really similar to how a college review session would be held. If you go to a large college and you attend a review session, most professors will stand up on stage, and if no one asks a question, they'll leave. Because they're not going to prepare a big presentation for you. They're not going to just go over everything from beginning to end. They will wait for a question, and if no one has one, the professor will most likely head on home. Okay, so we'll give it another minute. If you have any questions for Unit 3. No questions yet. Can you guys all hear me? Now I'm getting worried that maybe my internet's not working. Okay, thank you, Soleil. It's like, hey, maybe no one can hear me. That's a possibility. All right, I'll give you one more minute to think about a question. If there's anything you want me to go over. Actually, I can pull up the objectives and type them in here. Sometimes that helps if when you're studying and you can't think of what kind of, like, you can't even think, you're like, I don't know anything, I can't even think of a question. My recommendation to you would be go to your teacher or the AP College Board objective list and look through the statements that say what you should be able to do. And if you're looking at those objective lists and you're like, I don't know that one, that's kind of a good place to start, right? It's a huge way to figure out what you don't know. Okay, here, I'll give an example of an objective that I'm pulling up on the, okay, so learning objective number 1 from this unit is describe the properties of enzymes, right? So can you describe the properties of enzymes? If you can, great, move on. If you can't, dig into that objective. Thank you, Aiden. We have a question. Okay, so Aiden says the enzyme has a specific shape for the substrate. Yes, absolutely. So can you guys remind me, what is the macromolecule that makes up enzymes? We think back to macromolecules. We've got carbohydrates, lipids, nucleic acids, and protein. What is an enzyme? Good. Thank you, Soleil. It's a protein, right? So think way back to the very beginning of AP biology. We talked about biochemistry. Proteins are made, right, from the information in DNA, right? And so they're made from where DNA is read, and then it's an RNA molecule, and then the ribosome recognizes the RNA and pulls in amino acids, right? So it starts off as this like primary structure, the string of amino acids, and then those amino acids interact with one another to fold. So try to think back to protein folding, right? The protein starts to fold, and then those folds and interactions will give it its shape. So Aiden, enzymes are a protein, and so their shape is everything. Okay, their shape is everything. Okay. The substrate will fit it, right? It goes into the active site. So if you think of like this picture I have up here, the substrate is, you know, cartoon shown in like a semicircle and a triangle. That actual shape would fit into the active site of the enzyme. So if the enzyme changes at all, it's not going to function like it should. Okay, Soleil, will we need to know about C3, C4, and CAM plants? So as far as those, let me look into the AP College Board, because, you know, some teachers are kind of old school in a way where they would want you to know about those. But looking into, I'm looking at photosynthesis right now on the AP College Board course and exam description, the brand new one. It does not list C3, C4, and CAM plants as a learning objective. Okay, that does not mean that your teacher won't test you on that. Okay, so there's a difference between what your teacher might test you on, because you know, there's things I test my students on that I think are very important. They may not be on the AP test, but the AP College Board might give you a question that is about a CAM plant, and it'll tell you everything you need to know, and you may have to compare it to a normal photosynthetic plant. So I would check with your teacher on that one in terms of what they will test you on. For those of you who are maybe popping in and out, we're just going over any kind of questions that some of you might have in terms of photosynthesis, cellular respiration, and enzymes. Okay? If you have a specific question, ask away, and then I can kind of dive into it. Fructokinase? Oh, Diana. That is so beyond the scope and sequence of AP biology. You would never need to know that for the AP test at all. The phosphofructokinase is completely out of the scope of the AP test. That'd be something they wanted you to know like 10 years ago, but not going to be on the AP test. And Soleil, your question. Soleil asked, what is the action spectrum of photosynthesis? Are you talking about the absorbance spectrum? Did you mean to say absorbance? I'll give you a second to let me know if that's what you meant. Because if you're talking about the absorbance spectrum, I actually have a free response question that we could talk about, and I could show you. Here it is. So I'll give you a second to respond to me. Let me know if that's what you're talking about, is the absorbance spectrum. Maybe? Okay, I will put in a little picture into the PowerPoint real quick. So give me a second. Let me just insert this image. And then we will talk about the absorbance spectrum because it's a really good one to know for the test. Let me make it nice and big. Okay, here we go. All right. Presenter view. Let's make sure that works. Okay, so hopefully you can see the image. This is from one of the AP Biology free response. Gosh, what was it, like 2016 or 17? What year was it? 2013. So this was the first year after the big change where they made AP Bio less about memorizing and more about thinking. Okay, so these are two graphs right here measuring the relative absorbance that the plant is having. So how much of a certain wavelength of light is being absorbed. So if you haven't taken physics yet, we think about white light from the sun, right? It can be split up into different wavelengths, right? When we think about what the rainbow is, Roy G. Biv, every color has a different wavelength of light, right? So in terms of like an actual wave, right? We think of a wave, wavelength is how close those peaks are to each other, the crests. So if you look at this chart right here, it says violet has wavelengths that are shorter, right? So those are like higher frequency waves, higher energy. And then down below is red. So that's more in the 620, 750 range. So reds are actually really lower energy colors and also, just larger wavelengths. So this question was telling you a little bit about this green algae, I think, and then there was this bluish bacteria that both were capable of doing photosynthesis. So what it was trying to get at, can someone type in the chat box, when we think of photosynthesis, what is the organelle responsible for photosynthesis? So what's the organelle that's responsible for photosynthesis? Chlorophyll is the pigment responsible. Chloroplast. Yeah. Okay, good. So if we know that chloroplasts are the organelle responsible for photosynthesis, and then we dig even deeper in terms of what is inside the chloroplast, that's where Soleil was on the right track. Chlorophyll. Okay. What color do we associate with chlorophyll? Green. Okay, good. So we associate green with chlorophyll. So here's where a little bit of physics comes in and students might get confused. So if we look here in terms of a green wavelength of light, so the color green can be at a wavelength between 495 and 570. So you have to think about, well, what makes something green, right? If I'm wearing a green shirt, is it because the light that's reflected off is absorbing green, or is it reflecting green? So if my shirt is green, let's pretend it's green. Is my shirt absorbing green or is it reflecting? Yes, you guys all took physics, right? Maybe. Yeah, so it's going to be reflecting green. So if you look at these two graphs, if the y-axis is absorbance, and I'm asking you, hey, which of these graphs is showing me a green photosynthetic organism? We got to look. Okay, well, graph 1 has high absorbance at, between, it looks like around 550, right? Between 500 and 600. Okay. Graph 2 has almost no absorbance between 495 and 570. Okay. So graph 2 has no absorbance at that green wavelength. Yeah, good. So let's say the question in this in this free response question, asked you, which organism is the green one, which one is the purple one. Okay. And then, right, so we would say graph 2 is going to be your green photosynthetic organism. Okay. Most people, I used to give this to my students every single year, and they would all jump to graph 1 because they'd see a peak around 500 and they'd go, oh, look, it's a peak of the exact same wavelength of green. But we're not looking for absorbance, right? We have to think back to the basics of green means reflecting. So if you're reflecting, you are not absorbing green. So that's why graph 2 would be showing us our green pigment. So this would be responsible for a plant that has a chlorophyll, or high amounts of chlorophyll doing photosynthesis. Okay, so hopefully that kind of makes sense. And maybe that's where you were going with your question, Soleil, in terms of needing to know the idea of, absorption spectrum of photosynthesis. An extension to a question like this, I could see them saying something about, how we get different things than chlorophyll. We get like carotenoids and other pigments that are responsible for doing photosynthesis, like in the fall, right? And so we think of like how leaves change colors, and that's because we've got different pigments who are now taking over to do photosynthesis. So they're still able to absorb light, but they just don't do it as well as the green chlorophyll does. So I could see an extension of that where like it could say, hey, draw a graph of relative absorbance over wavelength for a plant that's in the middle of an autumn, right? And so you'd want to show, you would show a peak at green rather than a low trough for green. All right. Awesome. Oh, I forgot to go over my goal for you today. I'll do it right now. So I have 2 goals for today that aren't really content specific necessarily. They're more personal. So the first one is to strengthen your ability to recognize what you know well and what you don't. And then the second one is to advocate for your own learning. So that means ask questions. So if there's something I'm talking about and you're like, what is she saying? Like type it in, say something and be like, I don't understand what you meant by this. Because, it's Monday and I'm human and I might say something that doesn't sound right to you. So if you're confused about what I mean by absorbance and reflection, don't be afraid to ask the question. It is okay. This is why we have this platform. Because instead of just watching a video and not being able to interact with it, you can watch this and actually ask me what I meant by whatever word I said. Oh, yay. Okay, we have a question and 2 people have voted for it. So Aiden, thank you for asking this question. So how can the active site lower an activation energy barrier? Okay, so I'm going to kind of reword your question to what I think you're asking. So if we think back to enzymes and how they are, let me go back to this picture actually, because it does show the activation energy. Here we go. So if we think about this picture on the top right here, so I've got energy on the y-axis and time on the x, and it's showing you in blue with enzyme, it's requiring a lot less energy to get to my products. And then without the enzyme, we've got to get over this huge red hump in order for the same reaction to occur. So let's pretend the reaction is to split, let's see, what do I want to split? Hydrogen peroxide. So I have H2O2 and I want to split it. And so the enzyme that's in your blood called catalase can actually split that. So without it, though, the body would have to work really hard to split hydrogen peroxide, H2O2. Okay. So the way that an enzyme lowers your activation energy is it basically, so because your substrate, so let's say the thing you're trying to break up is H2O2, it's almost like, how can I explain this? Think about like, you know, when you're cold and you like rub your hands together to get warm, right? So you're like creating heat because you are doing motion. Almost think about like when the substrate, the thing that we're trying to break apart, when the enzyme is interacting with it, it's kind of messing and moving all those bonds around and increasing the kinetic energy. And so then that will allow for those bonds to break apart. So instead of it being like a stable molecule, the enzyme is allowing for that break to happen much more quickly. So the energy that it takes to activate that change, that breaking of the substrate is lowered because the enzyme catalyzes it and it says, let's speed this up. And so you get that, that bond between substrate and active site, and then that will make things go more quickly. Okay. Any other questions? And if not, could you explain? Yay! Okay, so Diana has a really good question about competitive and non-competitive inhibitors. So I want to actually move forward into a free response question that I have ready for you guys. That is, here it is. Okay, good. So we'll kind of work through this free response question together, Diana, and we'll talk about competitive and non-competitive inhibitors. So when we're talking about inhibitors, for those of you who are like, what are they talking about? So with enzymes, if you remember, maybe you guys did a lab where you heated up an enzyme and you measured product to see, well, how did heat affect it? How did changing the pH affect it? How did increasing substrate affect it? Substrate concentration. Hopefully you did a lab similar to that, or at least looked at data for that. So those are ways that we can affect the efficiency of an enzyme. But there's another thing that can be thrown into the mix, is that we can actually compete with an enzyme. And when I say we, I mean living things and cells, right? So like the actual organisms and cells that have enzymes that need to do jobs. So if you need to zoom out for a second and just remember what an enzyme does, enzymes are in all of our cells, and each enzyme has a specific job, and it helps to make things more efficient, right? So even like zooming out to like, okay, hey, what did you have for breakfast this morning? If you had a bagel, when you started chewing that bagel, there's an enzyme in your saliva that starts to break up those bonds of the food. And when we break bonds, we can use it for energy, right? And so the idea of breaking bonds, the bonds are what holds the energy. And if you're able to break them, you're able to harvest the energy and make it into something usable like ATP, right? Okay, so anyway, let's look at this free response, and then we'll talk about inhibitors and how things can affect an enzyme. So we've got a graph here. It says the graph shows the initial rate of an enzyme catalyzed reaction at different substrate concentrations in the presence of a constant concentration of the enzyme. So A, connect the primary structure of the enzyme to its overall shape. So we kind of already talked about that in the beginning, and how we talked about how enzymes are proteins and how that primary structure is the sequence of amino acids that was dictated by the DNA code. So if there's any kind of mistake in that, it'll affect the structure. But the idea is the primary structure is set by the code in DNA. And so that order of amino acids and the biochemistry of each amino acid, whether it's acidic or basic, whether it's hydrophobic, hydrophilic, will dictate whether or not it folds this way or that way. And that it's folding, right, from primary, secondary, tertiary structure gives the enzyme its shape. So that's A. We'll just kind of skip past that. So B says, predict the effect of adding a non-competitive inhibitor to the reaction mixture on the rate of reaction at a high substrate concentration. And I want you to support your prediction by describing how the non-competitive inhibitor affects the structure and function of an enzyme. So my next slide has a good picture that we can talk about. Here we go. So the reaction at the top, it's just showing what a normal reaction is. So don't really worry about that unless you need to reference it. But the idea is you've got a substrate that the enzyme will bind to, and the enzyme will allow the two products to be formed, or however many products. So with inhibition, there's competitive and non-competitive. So competitive means this inhibitor, this molecule, literally and physically blocks the active site. So it is competing for the active site. So think it's competing. It's like they both fit, right? They both fit in the same thing. So like if you're in a sport, and let's say you're the soccer goalie, right? If there's two goalies on the team, only one can play. right? Only one can play at a time. So similarly here, there's only one active site. So either the substrate is getting in there or the competitive inhibitor is getting in there. Okay, so hopefully that's making sense so far with my sport analogy. I'm sure you can come up with your own, and I actually encourage you to do so. Coming up with your own strange analogies to make sense of this stuff, especially because it's all micro. Like I can't see this happening. I can't physically watch, an enzyme connect or not connect. So come up with your own analogies, and it makes a big difference. Okay, so competitive ones would bind to the active site, and then, the substrate would never be able to bind there, okay? But here at the bottom, it's showing you where the inhibitor is binding to a site that's not the active site, okay? So it's binding to a site called the allosteric site. So not the active site, the allosteric site. So it binds there, and then that binding, you can see in this picture, the active site used to be a triangle, and now it's kind of like part of a hexagon, okay? So the allosteric inhibitor changes the chemistry, changes the shape of the active site, and so the substrate just can't even bind there anymore on the ones that have been affected by the inhibitor. Okay, when a non-competitive, because they're not competing for the active site, when a non-competitive inhibitor comes in, there are still some enzymes that haven't been affected yet. It's not like let's say I have 500 enzymes in a solution or in a sample, right? And a non-competitive inhibitor comes in. It doesn't mean that every single enzyme is going to be fully saturated with that inhibitor, right? So imagine all these molecules are moving around. And so when a non-competitive inhibitor comes in, there's still plenty of enzyme that can work with your original substrate, right? Because if you look, if they're not bound, the substrate and active site are still there, right? With competitive, those active sites would be totally blocked. And so it would be more difficult for your substrate to bind. Okay, so that's first thing. It's just being able to talk about competitive versus non-competitive. Drawing this out also really helps, just something similar to this, make your own shapes, make your own pretend substrates, and talk yourself through. Like, this is how it should be, but then this is how it can be inhibited. Okay, so my next picture is just my own hand-drawn picture showing what a normal enzyme would look like in terms of, full substrate concentration. Okay, Aiden, we'll get to your question in a second. So the normal enzyme would be, just it reaches that saturation where every enzyme that's in the cell or solution is used up, right? And not used up, is saturated, right? Is filled with substrate. With a competitive inhibitor, you can still eventually get to that saturation point. But with a non-competitive inhibitor, because it's actually, changing your active site, you can only get to about half of what you normally would. Okay, so competitive, you still have enzyme that's able to bind with your substrate. Okay, so it's like each one has an equal opportunity to get there. Eventually, your substrate will be able to. It's more like a statistical thing where your substrate would still be able to get to saturation point of your enzymes. But the non-competitive inhibitor, because of that actual change to the active site, you can only get to about half in terms of getting to that saturation point. Okay. So again, Diana, if you get tripped up between competitive, non-competitive, think about like sports. Competitive means you're both playing for the same position. You're both wanting that same position. So the two goalies, only one can be in goal at a time. So with competitive, you're competing for the exact same spot, the exact same active site. With non-competitive, you're not competing for the same active site. You're competing for just the enzyme in general. Aiden is asking, what is the purpose of competitive inhibition regarding enzymes? So organisms can use this as a way to regulate enzymes. So for example, if there's something that is breaking down too rapidly, or if it's breaking down too much, the body can throw out, and we'll get more into this when we get into homeostasis, but the body could put out, sorry, my computer keeps giving me notifications about the stream that's already happening. Anyway, so it's more about like regulation. We should be able to get more into that in the next unit because it's all about cells and cell regulation and communication. We get way more into like the biochemistry of that. But plainly speaking, it's a way for cells to stay in check and not just allow full substrate or full enzyme saturation. Sorry. Great question. Okay. I'm going to move along to the next free response question. If there's something that comes up in your head, go ahead and write it in the ask a question button and we'll get to it. So this free response question is about ATP being, well, yeah, it's about ATP, but basically this is an example of a free response question that you would see where there's data. It's going to ask you to graph it and justify some stuff that you're seeing. So I know that you probably don't have graph paper with you right now. So this is hopefully, you know what I'll do? I'll share this presentation with you guys. So even if you're not a Fiveable member, I'll do it at the very end before we all leave. Even if you're not a Fiveable Plus person that gets access to these later, I'll just give you the link today so that you can have these pictures. But anyway, so basically, sorry, I'm like losing my concentration. So I'll just read you the prompt. So researchers are studying the relationship between glucose concentration, it's our first column, oxygen level, and ATP production in one type of mammalian cell. Cells are isolated and cultured in a growth medium, either containing 1.5 millimoles glucose or 25 glucose, and at oxygen levels that varied from 0 to 21. The researchers determined the concentration of ATP per cell under the different conditions. The concentrations are shown as relative to the maximum ATP concentration obtained when cells were cultured in the presence of 25 millimoles of glucose and 21% oxygen. Okay, so first the question of the prompt was, we'll just ask first, let's look at this and answer, what is the role of oxygen in the production of ATP? So actually, my pre-question to you is, are we talking about cellular respiration here, or are we talking about photosynthesis? Go ahead and type it in. Is this kind of question talking about photosynthesis or cellular respiration? It's talking about glucose concentrations, percent of oxygen, and ATP. So are we talking about cellular respiration or photosynthesis? Thank you, Soleil. Yeah, cellular respiration. I know that might seem like a silly question, but I've had a lot of students in the past because both of these reactions have lots of steps, right? It's cell mediated, it's nice and organized. They both have an electron transport chain. They both have ATP synthase. And so students will think about it and be like, I don't know, like, you know, and they both involve oxygen and glucose. And so, you know, just wrap your head around why, you know, this would be cellular respiration, right? Concentration, glucose is being broken down, oxygen is being used, and ATP is being made. So just think about that. Okay, so second question is the official question. What's the role of oxygen in the production of ATP? So go ahead and type your answer in. What's the role of oxygen in the production of ATP? I'll wait. So what is the role of oxygen in the production of ATP? Okay, so if we think back to our, oh, thank you so much. Final electron acceptor, yay. For those of you who are having a mind blank of what are we talking about. So with cellular respiration, remember we've got these three main parts. We start out with glucose being ripped up, so glycolysis, glycolysis. We make a little bit of ATP, but most importantly, we're grabbing electrons. So when we start reconfiguring molecules and breaking them up and moving bonds around, the whole point is to take electrons, because electrons are going to be put through this electron transport chain to do some work for us so we can make lots of ATP. Okay, so if we look at the very end here, this is just like a cartoon model of cellular respiration. At the very end, where we have oxidative phosphorylation, that's your electron transport chain. This is where the most ATP is made. And if we think back to what the electron transport chain looks like, okay, the whole point of the electron transport chain is to take energy from all these collected electrons, right? So we've broken up glucose, we've changed pyruvate around like crazy, right? We've released CO2, we've moved all these things around, but really what we're doing is filling electron carriers. So your NAD and your FADH have been filled with electrons, and so protons too, right? That's why they're called NAD, but then when you add to NAD plus, and when you add to electrons, they come with a proton too, right? So we think back to chemistry. That's why I hopefully took chemistry last year or recently. Anyway, the whole point of that is when your electrons are falling down, we're able to harvest that energy, and it's pumping hydrogens across the inner mitochondrial membrane. Because of that, once there's a gradient, all those hydrogen ions can flow through the enzyme, which is it's an intermembrane enzyme, ATP synthase. And when that flows through, it actually spins and phosphorylates ADP and makes it into ATP. Okay. The thing that's pulling electrons through this entire chain without it is oxygen. So oxygen is super electronegative, which means it's loves electrons, it wants them, it's kind of greedy. You have these electrons that are being pulled through. It's almost like, I imagine it being like a vacuum, like oxygen is sitting there with a vacuum and like sucking all the electrons and pulling them toward it. So oxygen is the reason the entire electron transport chain works, and is the reason that ATP is made in this step, right? That's why if you hold your breath and you can't have any oxygen, your cells die. Right? They use ATP like crazy. So if you stop breathing, if you don't have oxygen in a cell, it can't make ATP and do its job. So without oxygen, it'd be in a rough shape. Okay. So you would get a grid similar to this. So I'm going to actually go back to the data real quick. Where's the data? Wait, back up to the data. Okay. So if we go back to the data, you might be a rock star at graphing. I don't know. However, when there are 3 columns of data, I know my students start to be like, what do I put on the X? What do I put on the Y? Oh my gosh, I'm freaking out. You kind of just take a step back and look at it and think about what makes sense, right? Even if you have to kind of draw a sample graph on the side and be like, okay, well, if I put concentration of glucose on the X, I only have 2 options, right? It's either 1.5 or 25. And that seems a little funky, right? Like you could, I guess, do 1.5 and 25, but then you have 4 data sets per thing, okay? So you could try it and see how it looks, but won't look great. And then you look at percent of oxygen. It looks like it's nice. It's changed from 0 to 5 to 10 to 21, right? So this is a different growth condition. And then concentration of ATP per cell is a measured thing. So I would put oxygen on the X, ATP concentration on the Y, okay? And focus in on just one line for 1.5 millimoles of glucose. Okay, so this is where you're going to need a legend. Here, I'm going to do like a sample graph real quick here. I should have made it dotted, my bad. Okay, so I'm going to show you, where am I? Okay, so here's like my sample graph, okay? Oxygen would be on the X, ATP would be on the Y. I know my picture's flipped, but you're going to have 2 lines, right? And so your legend will show, oh, this line that's straight is for 1.5, the other one is for 25. I know I'm talking really fast, but that's it. That's what I would do. Okay, and again, I'll share this with you at the end today, so you can do this on your own and give it a try. Okay, so I would graph it. You get 2 groups, so 2 lines of data. The next question would maybe ask you, describe the relationship between concentration of glucose and ATP concentration. This is where if we have a slight difference in numbers, it's really nothing, okay? You could do like standard deviation and whisker bars and all that. But if you look at percent oxygen for 1.5 is 0.3, and percent oxygen for the next one is 0.4. If you look at the two comparisons of, 15? The glucose concentrations, 1.5. So comparing 1.5 glucose concentration to 25, if you compare the relative ATP, they're barely different from one another. So don't look into it and be like, oh, well, look, at 21% oxygen, this one's at 0.85 and this one's at 1.0. Yes, that's different, but look at how closely all the other ones are. 0.3, 0.4, very close. 0.65, 0.7, super close. 0.85, 0.8, basically the same thing. So be careful with questions like this. If numbers are similar, don't be afraid to say, hey, there is no difference. It's okay to say that. You don't have to be saying that, look, this experiment showed us something miraculous. It's okay to say, actually, there is no difference between the two. Okay, I think some people are afraid of that when they look at the numbers and they're like, well, they are different, but not by a lot. So that's where your familiarity with data is really important, and be confident in knowing that, like, hey, if this is 0.8 and this is 0.85 and all the other ones are pretty similar, then just be okay with saying they're similar. Okay, last question, and then I'm gonna give you the slides and head out. Here we go. If a compound that binds and releases a proton is added to the growth median, predict what would happen to ATP production. Okay, so if we think about how important protons are, remember that picture, what would happen to ATP production? So think about that. While you think about that, I'll get the link to share this with you. I don't know if it's going to let me. Hopefully it does. So if there was something that stopped proton, or took all the protons away, what would happen to ATP production? Sorry, I'm trying to get this link copied for you guys. It would decrease. Yes, thank you, Soleil. You're still with us. Yeah, so if we're messing with the protons and all of a sudden all these protons are getting scooped up, then absolutely, we would never have that concentration gradient created. Maybe a slight one would be created, but not enough to make ATP synthesis spin like crazy and get ATP to be made. So you justify it. And when it tells you to justify, basically tell the proctor, the person who's going to be reading your essay, tell them everything you know about that part. Right? So if you're talking about ATP production, tell them everything you know about ATP production. Be as specific as you can. Use the buzzwords like concentration gradient and, ATP synthase as a transmembrane protein, and, use the stuff that you know so they know, what you're talking about. Okay. All right. So we're at 43 minutes. Try that link. Make sure it works. Okay. If you guys ever have any very specific burning questions about whatever, again, I have my Twitter. Let me show it here. I'm on Twitter quite a bit. I like interacting on there, especially if students have questions. So feel free to just direct message me on Twitter. It says request access. Why does it do that? All right, we're going to get this figured out. Okay. I don't know why it keeps saying that only people with Fiveable can do it. Okay, specific people can access. Oh, here it is. On anyone with the link. Got it. Done. Copy. All right, I'm going to try it again. All right, try this one. I have already sent this message. Okay, try it again. I've just changed it. So try that same link again, actually. I've just changed it to mean anybody with the link can access it. So try it again and let me know. Did it work? Oh, yay. Diana said she got it. Awesome. Okay, so again, guys, I know, oopsie. I'm sure we didn't get to every single thing that you need, but again, please, don't be afraid to direct message me on Twitter. I'm happy to answer your questions on there. And for those of you who have tests tomorrow, I wish you the best of luck, and we'll see you next time on here, okay? All right, good job doing work. We'll see you guys around. Bye.